Today I will talk to you about PBPK-compliant 4-organ chip for drug development. I'm a senior scientist at TissUse, and I will start the topic with giving you a little bit background, our motivation, why we are doing what we are doing, and also a little bit of the historic background, how the field is developing. So here in the a little bit of introduction, and then I will show you how our assay development looks like, and some examples of case studies, and an outlook, what we are expecting to come. So our motivation comes from the problem of traditional drug testing. It has dramatic failure rates in the clinical studies up to above 80%. And this is actually making a drug discovery, a new compound entity, very expensive to bring it to the side of the patient, starting from the preclinical studies, clinical studies, and to reach the patient. And we see the problem actually comes from the preclinical side, where we have basically using animals for testing the compound in a systemic manner, or to have a human system in cell culture, which is not systemic. You can imagine the animals have a different genetic background, different metabolic properties, which makes it quite in a high percentage irrelevant, could make it irrelevant. And this is the reason there's high clinical failure rate. So this is actually the point where we are expecting a paradigm shift by being able to combine human and systemic aspects of the preclinical testing together, which will be with the microphysiological systems, to make it more organotypic, miniature, human-like systems. And this would also allow the preclinical development, the drug discovery period much shorter, and then the clinical failure rate lower. And we would hope for a success rate, hopefully above 80% with the systems in use. So when we look back in the history of the microphysiological systems, we are talking about a roughly around a 20-year history. It's really a new field. And at the moment, there are already 360 research groups, more than 360 research groups working on this, with in just 20 years, more than 1,000 publications. It's really expanding. It all started around 2002 with single tissue microphysiological systems, really simple systems. And then it started to expand into, develop into multi-organ system co-cultures or single organ systems. And in around 2012, NIH had a very big grant to basically also establish the field. In 2015, it's HDMT. These are big grants who really pushed also the field forward. In 2016, there was the 14-organ microphysiological system to mimic human on a chip while in place. And now more and more we are many of the commercial companies, the research groups are trying to make organotypic and also feasible for fit for purpose testing adapted. So, and when we look into the microphysiological system providers that is available, there are basically 4 groups we can group them into. So these are either plate-based systems or chip-based systems. So in this case, some concentrate on single organs or multi-organs. So we have these basically 4 groups which we can group. The differences would be according to the assay, according to the purpose of what we want to look into. Each group has its own advantages and disadvantages. Some are more high throughput. This is one difference. Some has more data-rich endpoints, and some has more advantage on the barriers. So this is really going into the direction, what is the best fitting system for the test we want to do, or the interactions we would investigate in. And coming from that, so this is basically the hardware part. And then there has to be also the software part, in this case, the biology coming together. So the cell sources. When we look into the cell sources, there are also many available cell sources. If we start from the adult part, the adult donor, there are already available primary cells. We are able to use tissue biopsies, explants, organ-specific stem cells. One example widely used is intestinal organoids from human adult intestine stem cells. This is as an organ-specific example. The other option is immortalized human cell lines. So these were originally from an adult donor, and they have been immortalized. They have their advantages or disadvantages, all of these. And coming back to the, if you consider patient-specific genetic background or patient-specific studies, what comes very important is that studying one donor as an autologous source for drug discovery, because we all have different genetic background and different responses to the drugs. And in that case, induced pluripotent stem cells become very important as a cell source because we can reprogram an adult cell into more embryonic stage. And this embryonic stage allows us to redifferentiate them into many different types of the organs, many different types of cells. It could be a liver cell, a brain cell, muscle cell, and so on. And this allows us to establish different organs of the same individual from basically a single cell source. So these are available basically cell sources. And then basically putting these together would be an assay. We would consider what is fitting for this new compound or assay we would like to try, and then we start with the relevant cell sources. We need to understand the model the best. Is it fulfilling the requirements? For example, does it have the proper transporters for the uptake of this particular compound? Is the cell line fulfill our needs, or do we need to use primary cells, commercial models, biopsies? And then model preparation, sometimes no time, sometimes it takes up to 4 weeks, depending. And then everything comes together in the chip or the microphysiological system of use. In our case, we are able to do multi-organ cultures in the chip, and we are able to sample daily to also look for to monitor the organs This is in online monitoring of the tissues to look for glucose uptake, lactate production, LDH release, non-invasive molecular imaging, and daily medium exchange also allows us repeated dose exposure. So far we established long cultures up to 4 weeks. And endpoint of an assay could be, let's say, quite standard histology, qPCR, omics, metabolite analysis with LCMS, and so on. So it is similar to a laboratory experiment, a bit more detailed, more data rich. And it most of the time is much longer than, or is possible to make it much longer than a usual in vitro static experiment. In the case of assays we established to give a clue what is available, we did some single organ for bone marrow, for hair follicle. We have done co-cultures of liver and skin, liver and intestine. Examples are long, as you see in the slide. Here, what I want you to pay attention to is some assays aim for safety, some assays aim for efficacy, some actually aim for both. But also, in the case of a microphysiological system assay, we see the assays in 3 different levels. It's like a proof of concept, a newly established assay, let's say, an established assay that we know the capabilities of the assay, and the 3rd level is that it's ready for robust testing, that we know the functionality, we know the limits, advantages, capabilities of the assay. We are doing robust testing, its technology transfer, laboratory to laboratory differences are minimized. So this is a qualified assay. In this case, we also I will also then give you an example for in this case, one of the most complex assays. And this is the case study for studying intestine, liver, kidney, and brain. And our aim was to look for ADME, absorption, distribution, metabolism, and excretion. And we use some compounds that are actually known, some known compounds. And it was in a collaboration with the EU Horizon projects. And here, the chip for the ADME purposes we designed is PBPK model, so physiology-based, pharmacokinetic, compliant for organ chip. And what do I mean with that? We have the organs. There are planned spaces for the organs in the physiological order. Here, for example, we have the intestine-liver portalway mimicked. The flow rates of the organs are proportionally placed, and also we have 2 different circuits, one for blood circuit and one for the excretory circuit, urine circuit. And then in the interface we have the kidney cells in place. And this was the design of the chip to be able to mimic absorption through the intestine, distribution with the microfluidic, and metabolism in the liver, excretion via the kidney. Here you can also see some preliminary results we have. We used the primary liver microtissues that's commercially available. And a primary commercially available intestinal model, this is an intestinal model, and then iPSC-derived glomerular and proximal tubular cells. As the tox target, this was our tox target in this particular case study. It was the neuronal spheroids from Biotalento. So it was not particularly for toxicology. So our aim was looking for toxicology. Brain was it is one target, either for showing seeing the effects or seeing toxicology. So basically one organ in question to see the effects. And I want to show you how the assay looked like and how the chip looked like here, a bit more, I think, giving you some better idea. And this is the blood circuit where we have the intestine for oral application. And also to mimic systemic application, we basically applied the compound directly into the flow. So then it directly went into without any barriers. So this is also one challenge. We compared like systemic uptake versus oral uptake, if we can mimic the uptake at all. And here in the excretory circuit, we have both different types of cells. And the the oral or systemic drugs were applied repeatedly, so for 7 days. So it was a repeated dose application. And when we were designing the chip, what we took into account for the PBPK approach, we want to make the chip possible to model for the to have some in vitro in vivo correlation, and for that we have to take into account which channel with which flow rate, which organs, in which order, and then also next comes the compartment. When we look into the compartment, let's have it as an example, the blood like intestine. Is it a barrier model? Do we need to take into account the cellular barrier? In this case, there's the flow coming in and out. There is the active transport and passive transport, diffusion, and what would be the binding kinetics to the plastic? Because we are not in the human medium, we are in a plastic medium. There's organs in it, but there's the flow. The microfluidics are made of plastic. We have to take that into account. And of course, protein binding. So these are the criteria we took into account for this chip. And to show you some results for the functionality of kidney in this case, is that here we are looking into the proximal tubular part, and we looked into albumin uptake, and we see at day 0, at day 7, at day 14 that the albumin uptake is steady. There's some changes between the chips, but it's steadily there. We applied haloperidol. This is also a compound for the basically used as an antipsychotic drug, and one of the model drugs. And we, as a negative control in the proximal tubular part, we actually used HACAT cells. These are epithelial cells of the skin, and so they should not take the albumin. And indeed, we have negative control here, and we see a good uptake in the proximal tubular cells after 14 days and after the compound application. What was nice to show that over the time of actually this is 3 weeks for the kidney cells, that in the blood circuit, so this is the blood circuit, we are looking in here. Blue is control, red is haloperidol, and we see albumin values in the blood circuit. And here in the kidney circuit, we have no human albumin as we see here, and there is the barrier function stably present over the time. Especially the first week is the time we give to the organ to establish itself, and after the barriers is formed, there's basically a steady barrier function we observe. And looking into the metabolic capacity here, we applied haloperidol systemically in a physiological concentration, and we see the haloperidol metabolite being formed after 14 days of viable co-culture. And one of the challenges in the 4-organ system is that finding a common medium that basically keeps all the organs happy. And this is something we achieved. And then what we also discovered is that the liver is able to metabolize. And for haloperidol, there was no excretion we could detect, which is also actually physiologically relevant because it's about 3% that should be found in the urine. So I think it's okay with that concentration. Okay. And now we are looking into the carbamazepine. This is another model drug we used to basically administer it over the intestine to mimic oral uptake. So this is what we found is that we looked into the intestine blood uptake and then excretion for the blood kidney functional barriers if this is established. And indeed, we see that carbamazepine uptake was shown as expected about 80%, and we could see the metabolite forming. Also, we could find the urine circuit with the parent compound, the carbamazepine and its metabolite. So this is a very good example of ADME on a chip, so that we could show the uptake, distribution, metabolism, and excretion. What is also interesting in this case with what we observed with the carbamazepine was organ-specific toxicity, early toxicity. So with the carbamazepine here, we look into the similar data as the albumin in the blood circuit. We see the control versus carbamazepine, and in the kidney circuit, we also see the barrier integrity is kept, that the human albumin is not in the excretory circuit or very low amount. However, we saw increased levels of AST and increased levels of LDH in the kidney circuit in comparison to the blood circuit, in comparison to control, and also in comparison to the blood circuit. So this was really the toxicity we observed, or increased LDH we observed in the kidney circuit. And so this concept we also took further to study in the autologous system. As I mentioned before, if you want to go into personalized medicine to be able to mimic all of the organs of the patient on the chip, then we also need to use basically iPSC cells to be able to have them from the same donor, because when we are using primary cells, this is really not possible. Okay, so what we then studied to make the whole chip autologous, we took the IP. We have basically iPSC bank in place. We differentiated a single donor into different organoids: intestinal for the intestine, for the liver, for the neurospheroids, and blood-brain barrier, and also for the kidney cells. And in terms of the blood-brain barrier, what we did was using the blood-brain barriers to face the blood circuit, and we put neuronal spheroids on the opposite side inside the insert to form, let's say, the brain, to form the brain inside and have the blood-brain barrier on the opposite side. Yeah, so if we look into the characterization of these cells, we formed first intestinal organoids with the gene expression relevant to their transporters, tight junction barrier proteins, and then we basically formed these intestinal organoids. We dissolved them and then formed them as a barrier model on an insert to make it into basically a barrier model. We looked into the epithelial resistance to understand the barrier properties, and yeah, and the diversity of the tissue was actually quite well. And the next model in the characterization was liver. We looked into the metabolic capacity of the liver. So, okay, we first differentiated the cells from the iPSCs and then looked into we characterized them for their expression. And if there's albumin expression, for example, if there's CYP3A4, one of the enzymes, liver enzymes, and we also induced if we could induce it, the enzyme expression in these liver models. Yes, there is inducible gene expression. When we apply the compounds, we can induce gene expression, but liver models are not at the maturity level of a primary liver. This is something we have to keep in mind to improve. And iPSC-derived hepatocytes are really hard to mature into a primary level. So this is a big challenge also in general in this field. Okay, next is iPSC-derived kidney cells. And what we did was differentiating into tubular cells and podocytes. Podocytes are the cells of the glomerulus. And tubular cells are a mix of proximal and distal tubular cells. And we also challenge them for their functionality, if they are doing albumin uptake. And we also could show dose-dependent albumin uptake for podocytes. You are seeing this on the very right side of the slide, and also both podocytes and tubular cells. We looked into the barrier integrity. We also looked into their marker expression. They are quite robust in this case. And we also challenged them with the OCT assay. And this is, we applied cisplatin-induced nephrotoxicity. And there, we use cimetidine to inhibit the OCT. This is a transporter. And the OCT inhibition basically prevents tubular cells from toxicity. And this is something also we could show. So coming to the neuronal model, or the blood-brain barrier model, we first established the brain microvascular endothelial cells to form the barrier. And we looked into their, so endothelial cell markers, their tight junction, their tight junction expression, tight junction proteins. And we also challenge the system if their presence or absence changes for example, glucose and albumin transfer. And very clearly the absence almost balances the glucose or albumin transfer in the compartment, and so they show their barrier integrity. And we also could show with compound relevant selective compounds or medications already in the market, atenolol and propranolol in this case we use. One actually is supposed to freely pass the blood-brain barrier, which we could show in the right side of the slide. And also atenolol, for example, does not supposed to cross the blood-brain barrier, and we could show this difference in the presence and absence of these cells. Okay, and what we did also was then placing, after characterizing the blood-brain barrier, we also then placed these neuronal spheroids in the inner side to make it into the full brain model. These spheroids are also actually possible to directly use them without the blood-brain barrier, depending on the assay we want to achieve. But yeah, we also actually characterize these neuronal spheroids. They are quite robust, I have to say. In general, iPSC neuronal differentiation is something quite robust, and we also produce actually quite good spheroids. And yeah. What was interesting to study was when we put all these in the autologous system, when we challenged the system with the model compounds, which I already mentioned some data before, but this is basically all systems combined together to show what is the uptake of these, or the differences of these compounds. Is it as we expected? As the pharmacokinetics is as we expected for the atenolol. We see the law, and then in comparison, okay, here we are looking into the brain passage, blood-brain barrier passage of the compound. How much did we detect in the kidney circuit? How much did we detect in the circulation, and how much was taken up from the intestine? And also for the carbamazepine and the verapamil, we see the differences. And in this case, for the autologous system, we were also able to deliver 4 organs from a single donor in place. And again, a 14 days long viable culture with a common medium. And we also could show the differences from the biological barrier uptake and the barrier integrity and excretion. So this is the second basically assay we established. So now I want to come to the point of the outlook, where we want to go from this. The outlook, our aim is to basically test the patient without needing to test the patient, to have the human and systemic complex system in place. And if you have a qualified universal physiological template in place, then we can also use it as a systemic testing for the compounds which we have no clue about what we have to expect. And eventually, if you want to have a diseased patient on a chip, we then, without the patient becoming sick, we can, for example, test for the metabolic disease on the chip. So basically make the chip sick before and then treat it or heal it and see the effect only on the chip and the effect of the new drug. So for that, of course, we need to expand the cryobanking to have the genetic diversity, different organ differentiation. So these are actually already done. The level of different organ differentiations are more or less in place in also throughout the literature. So many groups are working on iPSC, the pluripotent stem cell differentiation, and proper organoids. So differentiation of the organoids, and basically we are at the level of establishing the co-culture feasibility of these cultures in place to have the real human systemic complexity to come to the next level of complexity, and come to the level of human on-ship trials. So this would be really the ultimate level where we need to reach. And imagine having the genetic background of cell banks, and then a healthy, and in this case it's a type 2 diabetes trial in place, having a healthy and diabetic patients in place, and testing with a different genetic background. Then we can have different new compounds where we don't know, and then also the compounds which are already in the market as controls and healthy controls, and to be able to actually show relevant data that would be possible to convert to be able to do in vitro in vivo extrapolation. Okay, with that, I would like to thank you for your attention, and I would like to thank our partners that we worked on this elaborate, complex project together. And I will be happy to answer your questions if you have some. Thank you.